Good evening, everyone, and welcome to the Fralin Biomedical Research Institute at Virginia Tech Carillion's Maurice Strauss Distinguished Public Lecture. I want to give my special thanks to Maurice Strauss for his support of this important monthly event that brings contemporary issues and leading world experts in science and medicine to our greater community. Before I introduce tonight's speaker, I'm going to put in a couple plugs for the upcoming events in this program. The next speaker on February 10th will be Robert Wachter, Distinguished Professor of Science and Medicine at the University of California, San Francisco, and member of the National Academy of Medicine, who will speak on the digital transformation of healthcare, finally a time for optimism. And 2 weeks following that, Dr. Carol Greider, Professor of Molecular Cell and Developmental Biology at the University of California, Santa Cruz, and recipient of the Nobel Prize in Physiology or Medicine, will speak on the role of telomeres in genome stability, cancer, and aging. And before I introduce tonight's speaker, I just want to remind everybody to type your questions into the Q&A, not into the chat, and I'll try to get to as many of those as possible at the end of the lecture. So now it's my great pleasure to introduce tonight's speaker, and it really is tonight for her, I should add, Dr. Sharon Peacock, who is the founding executive director of the COVID-19 Genomics United Kingdom Consortium that, by the way, first detected the Delta variant of COVID. As of this month, the consortium sequenced well over 1 million COVID-19 samples from across the UK. In addition to her leadership role at COG UK, Dr. Peacock is Professor of Public Health and Microbiology at Cambridge University. She earned her bachelor's degree in medicine from Southampton University and her master's and doctorate degrees from the Open University in Oxford. She was then a senior lecturer in microbiology at Oxford and served as head of bacterial disease research for the Wellcome Trust major overseas program in Thailand. And she has been professor of clinical microbiology and director of the Bloomsbury Institute at University College London. Dr. Peacock has published more than 500 peer-reviewed papers on pathogen genomics, the evolution of bacterial and viral pathogen transmission, antibiotic resistance, and a wide range of infectious pathogens. She is a fellow of the American Academy of Microbiology, the Royal College of Physicians, and the Royal College of Pathologists. She is also an elected member of the National Academy of Medical Sciences. Dr. Peacock was appointed Commander of the Order of the British Empire for service to medical microbiology in 2015, and in 2018 was awarded the Unilever Colworth Prize for outstanding contributions to translational microbiology. Prior to the pandemic, her research centered primarily on genome sequencing of pathogens to provide rapid detection and support of public health, to overcome antibiotic resistance and prevent outbreaks in clinical settings. She redirected her entire research group to the pandemic immediately when it started. Her major contributions to the science of infectious disease include genomic epidemiological analysis of antibiotic-resistant Staph aureus, the use of integrated genomic and epidemiological approaches to define hospital-acquired transmission of E. coli and antimicrobial resistance genes, and transmission patterns of MRSA in hospitals and throughout the community. Dr. Peacock is uniquely positioned to have critical perspective and insight into the current global pandemic, not only with respect to molecular virology of the virus, but also in terms of public health, policy, and the sociology of the world's current challenges in this regard. We are very fortunate to have her here this evening. Please join me in welcoming Dr. Peacock.
Thank you very much for that kind introduction. And just to say that I'm really delighted to be here this evening. Yes, it's 10:30 at night for me, but I have to say that I've had a tremendous day with scientific discussions throughout the day. So thank you so much for inviting me. I'm just going to share my screen now and get the slides started. So I hope you can see my slides now. And my talk today is going to be looking at SARS-CoV-2 variants in the context of the past, the present, and the future. Now, my crystal ball generally has about 6 inches of mud on it, so I'm not quite sure how accurate the future is going to be, but I'd really like to take you on a journey to think about what the present and the future might morph into. So my talk, I've already said that I'm going to go past, present, and future, but I'm going to start by looking at how SARS-CoV-2 genomes have really supported the pandemic response. I'm then going to move on to looking at viral evolution of the virus, really what drives evolution and what are the outcomes from that. I'm then going to give you a bird's eye view of the recent past in terms of variants, then focus on Omicron, and then finally what happens next. So SARS-CoV-2 genomes in the pandemic, and the genome that I'm attached to the most is the very first one that was generated way back at the beginning of 2020. Now, this was generated by Chinese scientists, and this was really critical in our development of vaccines. It helped us understand what this virus actually was in terms of characterization, but it allowed very rapid development of vaccines against the spike protein. That's the part of the virus that interacts with the human cells. Now, since then, there's been a great deal of sequencing done to really inform the pandemic and provide actionable information. In particular, we can detect new variants of concern worldwide as soon as possible. We can reveal patterns of these variants, how they're introduced into the country, and then track their transmission within that population. Of critical importance is that we continue to use the genome information to monitor effectiveness of vaccines and treatments. And that really supports novel variant-informed development of diagnostics, vaccines, and therapeutics. And what I would say is that before the pandemic, pathogen sequencing was something that a small number of people really talked about and got excited about. But I think that the pandemic has really led to this becoming front and center in many people's minds and being discussed as an important tool for the pandemic and beyond. Now, next, I'd like to talk about viral evolution, which is a critical point when we're talking about variants and their emergence. I'd like to start with this. So at the top, you'll see a figure of the SARS-CoV-2 genome. This contains around 30,000 points in the genome, or nucleotides. Now, what we're particularly interested in is mutations or typos in that genomic information in the spike gene that encodes the spike protein. Now, if you see on the bottom left, you'll see that there are several proteins that the virus has, but the spike protein, as I've already mentioned, it decorates the outside of the virus, and this is what attaches to our cells through a receptor and gets into our cells through that. So we're really focused on mutations in the spike protein gene. Now, this virus actually mutates around twice every month, and those mutations are random, and so they may lead to no effect on the virus at all. So a mutation leads to no change in the virus. The alternative is that a mutation could lead to a disadvantage to the virus in the way that its biology functions, and that's unlikely to be observed by us through sequencing. The third variety, and these are quite rare, are that mutations that actually give the virus an advantage. And under those circumstances, it's possible that virus will then start to expand and become more numerous compared to other virus lineages. So what we need to do with our sequencing is to spot the important variants as they emerge as soon as possible to inform our response to them. Now, we do that through escalation and classification of variants through a four-step process. The first is that we look at the genomes of the SARS-CoV-2 virus and we horizon scan. We're particularly interested in finding genomes that have mutations that we already suspect are important in changing the virus biology. They may step up to variants of interest if those variants look as if they're leading to a surge in cases or outbreaks or are associated with reinfection. And if we escalate that to a variant of concern, that means that there's evidence that there's a change in biology that impacts on our ability to resist infection or control infection. Now, the last classification is actually a variant of high consequence, which we haven't seen so far. And that is a variant, for example, that will be fully resistant to vaccination. Now, this slide really talks about possible outcomes from the genetic changes that I've talked about, where the genome is mutated. Now, it comes in four different versions. One is the virus could become increasingly transmissible between us. The second is that it could dodge our immune system so that it avoids a vaccine or actually reinfects us after a natural infection. The third is that it could alter disease severity, either up or down, so make a disease more or less severe. And these changes can also alter the readout from some PCR tests, which means that the tests still function, but it allows us to have a surrogate marker for particular variants. So variants of concern, I've put the definition here by the WHO, and these are increase in transmissibility or detrimental change in the COVID-19 epidemiology, or increase in virulence or change in clinical disease presentation, or a decrease in the effectiveness of public health and social measures to or available diagnostics, vaccines, and therapeutics. But actually, the things that have really we've seen emerge that have really made a difference to the variants of concern are increased transmissibility and immune evasion. It's likely that altered disease severity, either up or down, is unlikely to be a driver for whether a particular virus emerges. It's likely to be a bystander of those changes. So now I want to look at what drives the selection of variants of concern. This is important if we're to control their ongoing emergence. So the first I'm going to talk about is uncontrolled viral transmission and COVID-19 infection. Now, I've already said that the virus mutates once or twice a month when it's passing from one person to another. That doesn't sound very much, but actually when you've had many, many millions of infections in the world, that's a great deal of opportunity for the virus to mutate and for a variant to emerge. Now, if we look at the cumulative confirmed COVID-19 cases, we can see that there's a graph on the left which shows you the cumulative cases by a region. You can see here in Asia, excluding China, Europe, North America, and South America, etc. And globally, WHO report that on the 18th of January, there'd been more than 300 million cases confirmed of COVID and more than 5.5 million deaths. So thinking about drivers, we certainly can see that, unfettered infection could be an important driver for the selection of variants. My next is transmission in populations with partial immunity to SARS-CoV-2. Now, this is possible because the virus can be selected from different mutants for infection in people who do have partial immunity. And in that way, particular variants can actually bubble up to the surface to infect people in a mixed population, some of whom have got immunity and some of whom haven't. Now, what we know is that actually there have been 9 billion vaccine doses administered worldwide. However, that's not the full story. If you look at this graph here of the share of people vaccinated against COVID-19 in January 2022, and the dark green is people who are fully vaccinated and the light green is people who are only partially vaccinated, you'll see that there's great success in some countries, but huge diversity across the world. On a worldwide scale, around 60% of people have been vaccinated, but actually in Africa, that's much less. For example, in Nigeria, the rate of vaccination is around 6%. And actually, if you look at the share of people who've received at least one dose of COVID-19 vaccine, this graph shows that very starkly indeed. You can see in a line that trajects up for the high-income countries that a large number of people in high-income countries have been fully vaccinated, but actually in the low-income countries, around 10% of people have been vaccinated. Now, this isn't just or ethical on a purely humanitarian basis, but it's also not sensible to allow this to continue because countries which have a combination of transmission and infection and a lack of vaccination means that variants are likely to continue to emerge. Now, the third driver for selection of variants of concern is actually when the virus goes from a human into an animal where it can become adapted and changed for that animal and then go back into humans again. Now, this is a compilation of mutations arising following sustained human-to-human transmission or human-to-animal passage. It's actually a little old now, this figure, but it shows that mutations can fall into 4 different classes around the virus. But actually what I really want to do is use this figure to show how through passage in animals, different mutations can arise. And these animals include gorillas, dogs, cats, lions, mink, ferret, etc. And some of the variants that have arisen, for example, here, the N501Y, that's arisen both in man and mouse, we know is important in transmission. So we had an episode in Denmark where we saw how the virus went from humans into mink and back again. And this is something that we need to be very cautious and watchful for, because this is a way that the virus can actually change and come back into the population and become more transmissible, or potentially avoid our immune system, or have a change in virulence. To date, that hasn't been a major problem, but I think we do need to pay particular attention to it. Now, finally, another driver for selection of variants is prolonged infection in immunocompromised people. This is people who have an impaired immune system, who can't shake off the COVID-19 infection once they get it. Now, this is a slide of a study performed in Cambridge by Ravi Gupta's group, in which they followed a single patient who was immunocompromised and in hospital for over 100 days. Unfortunately, he died from his infection at 101 days. So what this group did was they sampled this patient and took samples which was positive for the virus on 23 different occasions during their treatment, and actually did deep sequencing. That means they sequenced so that they could tell whether there were different variants in that sample over time. Now, so you'll see along here, this is a timeline from day 1 to day 101. And you can look at the prevalence of mutations in those samples over time here on this particular y-axis. On this axis over here on the right, that is the CT value from a PCR assay. So looking at this patient's treatment, they had 2 courses of remdesivir, which is an antiviral treatment, initially when they were admitted, up to the first 54 days. Now, there was very little change in the virus detected whilst the virus was sequenced then. But then the patient had 2 courses of convalescent plasma. Now, that's plasma that's been taken from somebody who's had natural COVID-19 infection and has antibodies to the virus. Now, what we see when that patient has had that is that actually over time, we see a very dynamic shift in the virus, in that mutants arise with very specific changes in their genome. So you can see here that this particular virus has a mutation in D796H. And here, this particular virus has a deletion labeled here. So you can see these dynamic shifts over time. The patient has another course of remdesivir, another treatment with convalescent plasma, and then unfortunately they die. Now, this is important for obviously clearly for the patient and their relatives, but also the fact that these combination of mutations, these 2 in particular, the D796H and the deletion, had led to a reduction in effectiveness of the convalescent plasma. So this shows that continuous infection in a person is like the virus in a training ground. It's training to dodge the immune system and to persist in the context of human infection. Now, if we look at the alpha variant, which is the first variant of concern that emerged in the UK, in Kent, what you'll see is this is a phylogenetic tree. So effectively, there are a large number of viruses and their entire genome are aligned together to look for differences. And when there's a difference, you can see a step in this particular genetic tree. It's like a family tree. Now, the alpha variant was very striking because it had 23 mutations compared to the Wuhan, the original strain. And what you'll see here is that actually the alpha variant was clearly very different. And this is the first time we'd seen this actually when the alpha variant arose. And this is what drew our attention to it when it emerged in Kent. Not only did it appear to be more transmissible, but it actually had these very striking genetics. And this likely that this variant actually arose in somebody who had an immunocompromised body and a virus that actually had been infecting them for some period of time. Now, what that led to in the alpha variant was actually quite a striking different profile in the spike protein mutations. And so this is a kind of a figure of the spike protein with labeled mutations around the spike. And actually, what we're going to talk about later is Omicron, which is our latest variant that's circulating considerably. And if you look at the spike protein mutations, there are a large number, around 30 in the spike alone. And it's likely that this also arose in an immunocompromised person. Now I'm going to move on now to give you a very brief bird's eye view of variants in the recent past. Now this is a figure of variants over time in the UK from the start of the pandemic right up to the middle of last year. And the point of showing you this really is that there's a period when the variants that we saw I would call this the pre-variants of concern period, and then the post. So in this period here, right the way up to about November or December, what you'll see is there's each color represents a different variant, and not variants of concern. They're just simply variants of the virus, which are they're not able to outcompete the other. So they're all in there mixed together, attempting to compete with each other, but actually none of them being more biologically fit than the other. Now, in the summer of last year, we saw a bump in that we started to see almost half of the cases in the UK were due to this red lineage here, which is called B1177. We call this a summer holiday lineage because it first arose in Spain, and it then tracked across Europe and became a very common lineage in the UK. But in fact, on closer inspection, that was due to holiday travel and nothing to do with the virus. It was to do with our behavior. But the point in November/December in 2021 is really when that whole variant pattern changed. And what you'll see here is the alpha variant emerging and very rapidly replacing all of that diversity that all goes. And then we get the delta variant arising, which then sweeps away alpha and becomes the dominant variant. And so the pattern going into the future is very likely to be that we have a single variant that's outcompeting another variant. So just to look at these patterns of variants of concern, both in the UK and the US, in these slightly cleaned up versions of the graph, what you'll see here is a graph that actually goes up now till January 2022. And then again, it's normalized. So on any given day, this shows the percentage of variants on that day. And so the gray is the mixture of variants of concern that you saw in that color picture. But you'll see here that in the UK, we have the sweep of the alpha variant, and then the sweep of the delta variant in these green shades, and then Omicron coming in and taking over very quickly in a period of 4 to 5 weeks. Now, what about the US? Well, this has a very similar pattern. The gray block here is lots of different variants, very noisy genetics over time. But then the alpha variant actually is introduced into the US and becomes dominant, and then the delta wave. And now we see the Omicron wave coming in to the US. Now we have 5 currently designated variants of concern: alpha, beta, gamma, delta, and Omicron. Alpha is almost gone now, and beta and gamma only ever cause fairly localized or regional problems. And now we are facing the delta variant, which is a common, but now being replaced by Omicron. Now, really interestingly, we don't see one variant of concern emerging from another variant of concern. They're actually different. So what you see here is, again, it's like a family tree, but drawn slightly differently to the trees I've shown you previously. So this particular pink blob here is the original Wuhan strain of SARS-CoV-2, which in this particular figure is called 19A. You'll see here there's a division fairly early on into 2 branches, and one of the branches really doesn't go anywhere. So perhaps that variant wasn't particularly fit. But the other branch then gave rise to all of the other variants that we see circulating. We can see that this has a series of major branches, but this particular branch going up to the top has led to the alpha variant and also gamma and Omicron, and other branches have led to beta and delta. And so what's happening here is that mutations are arising that allow variants to have a fitness advantage, and then they're being selected out by their characteristics. This is very consistent with convergent evolution, where a characteristic is singled out because it has particular behavior, such as spreading more effectively or avoiding our immune system. So if we want to look at why variants of concern have things in common, then what we can see here is that of the 5 variants of concern, they all have increased transmissibility. And there's a stepped increase in transmissibility, one upon the other. So Delta was more transmissible than Alpha, and Omicron is more transmissible than Delta. Now, some of these variants actually have dodged the immune system. In particular, Beta, Delta, and Omicron dodged the immune system, in particular Omicron, which I'll come on to speak about more. But disease severity, as I've said, isn't a particular feature of it. It will certainly be important in understanding whether it's increased or decreased, but it's not a driver for the evolution and emergence of variants. So I now want to actually focus on variants and the present. Now, I want to go through a few facts about Omicron because actually this is our big story at the moment. So we know that Omicron is present in almost every country in the world now. It spread very quickly. And you can see from this map from the WHO from beginning of January, that the orange is the verified countries where Omicron's been found, and the hatch blue is under verification. And so this has swept the world. Now, I've mentioned that Omicron is transmissible. It's highly transmissible, actually. It has a significant increase in growth rate compared with Delta. And it has an increased risk of a close contact becoming a secondary case, by which I mean, if someone in your family has Omicron, then the risk of you becoming a case is actually higher than it was with Delta. In addition, if you have Omicron, you're more likely to infect more people than you would have with Delta. So it really is very transmissible. Now, why does it have this high growth rate? And it's probably due to a combination of factors, including immune evasion. So it definitely evades the protective immune system. And it could well have potential intrinsic increased transmissibility through its biology. Now, because of this clear growth advantage over Delta, it's rapidly replacing other variants that are circulating in other countries. So this is a map from the WHO showing how Omicron is replacing Delta everywhere, with faster replacements than ever seen before during the pandemic. Now, another feature about Omicron is that it appears to be behaving very differently, in that it appears to show preference for the infecting in the upper respiratory tract, by which I mean this figure here, so really above the vocal cords, rather than the lower respiratory tract, which is what the Delta variant would infect. Now, being associated with the upper respiratory tract might confer a transmission advantage independent of immune evasion. But actually, it's interesting that preliminary studies suggest that Omicron appears to have decreased ability to infect lung tissue, which might be why people infected with Omicron have less severe disease compared with Delta. So its biology is fundamentally different. Now, I've mentioned that it can infect the lower lung less, and it appears to be associated with lower severity and lower proportion of people going to hospital compared with previous variants. But there is a but. So the very large number of people being infected with it translates into a significant number of people requiring hospital admission, putting strain on healthcare systems across the world. Now, focusing on vaccines, vaccines protect against hospitalization, but actually less so against Omicron symptomatic infection. So all initial vaccine effectiveness estimates do show reduced effectiveness against infection and symptomatic disease compared with Delta. But it does show that current COVID-19 vaccines are providing actually very strong protection against severe disease and death, which is a very good thing. And preliminary vaccine effectiveness estimates show greater protection following booster doses rather than the primary series for most products. Now, I'd like to go on and show you some of that information in figures for the UK and for the US. What you'll see here along the top is UK cases of COVID-19 over time since the beginning of the pandemic. And what you'll see here is actually a massive peak of cases. And so we reached 200,000 cases, which is something we've never observed before around Christmas time. Now, you will also see that that peak is beginning to come down now. So it looks as if we are past the worst of our Omicron wave. If you look at the bottom figure, this figure shows deaths within 28 days of a positive test for COVID-19. And what you'll see is a decoupling between the number of cases that we're observing and the number of deaths we're observing. So in the first wave, you see a large number of deaths, and we had a paucity of testing, and so we don't have many cases. And then you'll see a much larger wave in the middle part of the graph. But at the end, despite having a very large number of cases, we don't have the same number of deaths. So there's this disconnect between the number of deaths and death. Now, that's also reflected in the number of patients admitted to hospital. And these are also somewhat misleading, in that the number of people going into hospital includes people going in because of COVID and people going in with COVID, i.e. they're going in with their heart attacks or their strokes, but they're also infected with COVID, but they're not going in because of their COVID. So around half of cases are going in with COVID as a coincidental infection. However, we mustn't underestimate the importance of Omicron in any country. The reason for that is that actually it takes a lot of people out of their work and their school, etc. And in the UK, we've had major disruptions from schools, from hospital workers who haven't been able to go into work, from train drivers. So we've had numerous cancellations of our trains, restaurants, etc. Now, what's happening in the US at the moment? Well, these are daily trends in numbers of COVID-19 cases in the US reported by the CDC, showing daily cases over time. And what you'll see here is actually you have also had, and I'm sure that you'll be more than aware of this, a massive spike in cases, the majority of which will be down to Omicron. And you'll see that the curve is just beginning to come down. So one hopes that although there's mixtures and heterogeneity in different states in the US, that you may be beginning to see the end of the Omicron wave as you know it. Now, I went to the CDC website to also get an observed and forecasted weekly COVID-19 cases in the US. And what the US are forecasting is that actually there will be a downturn in cases in the near future. And so that's consistent with what is being observed. Now, what about deaths? Now, your daily deaths in the US, according to reports from the CDC, also show that although you've got a major spike in cases, there hasn't been a major spike in deaths in the US. Now, again, I've gone to the observed and forecasted weekly COVID-19 deaths in the US. And what the CDC suggests is that there will be an increase in the number of deaths before it comes down again. And that is presumably because there's a lag time between people getting infected with COVID-19, developing severe symptoms, and then sadly, in some cases, dying. So at the end of the Omicron section in the current, I would say that the world in general is in a very stormy place at the moment, with Omicron actually going across the world and causing considerable trouble for people managing health services and also for the population as a whole. So what's going to happen next? Well, I'm going to cover three areas. One is what could happen to the virus next? What is its life trajectory, if you like? I'm also going to talk about how we consider the view of our future. So I'd invite you to sort of come along with me and think about how you view the future of Omicron. And finally, I'm going to talk about what comes after Omicron. So if we're thinking about possible future scenarios for SARS-CoV-2 as a virus, there are 4 possible outcomes that it could track to. The first is extinction. Now, we do have viruses that have gone extinct, and that includes the original SARS-1. That virus actually has become extinct, and that was because it was largely transmissible when patients were very sick. And so very stringent infection control measures managed to lead to eradication of that virus. That's very unlikely to happen with SARS-CoV-2 because it can transmit between people who don't actually know that they're infected. Furthermore, this virus is extremely transmissible. The second possibility is global eradication. An example of this would be vaccination for smallpox. So we've got rid of smallpox. But actually, the vaccines we're using doesn't eradicate infection or completely remove it. It reduces its severity. So global eradication isn't an option. The third possible future for SARS-CoV-2 is elimination through a zero COVID policy. This is where the virus is still around, but you don't have any disease because of your draconian measures that you take. Now, this is the policy being followed in some countries, including China. And I would say that this will be very difficult to hold in the longer term because at some point, you're likely to have to open up your country to maintain its or people's mental health, its economic stability, and all sorts of other reasons. So what we're left with is endemicity. What does it actually mean in this context? Well, it's possible that we're going to have to redefine what endemicity really means for this pandemic. But what I mean by this really is that disease is contained, is in a contained situation where immunity and precautions mean that disease rates are maintained at a particular or roughly particular level and an acceptable level that we can live with. So there's background of disease and possibly with seasonal spikes potentially over the winter. Now, a good example of something that's endemic is influenza, and this potentially could become endemic like influenza. To consider how we view the future, I've given you a series of facts and figures, and I think that how we view the future is going to really shape the social narrative and our decision narrative going forward. And I've given us 3 options about what happens next. The first is the optimist, the second is the pessimist, and the third is the fatalist. And these views are all mixed in. If you read the media, if you listen to politicians, if you look at how different countries are reacting, each of these are a possible outcome, and you may identify with one more than the other. Now, going ahead, the optimist would see that Omicron is mild, it's very transmissible, and it will infect billions of people in the coming months, but it's a natural vaccine and will provide an immune response against deadlier variants. And the optimist would think that the pandemic is almost over, the disease is becoming endemic, and we're going to reach a dynamic equilibrium with the virus, and we're going to learn to live with the virus. You know, the end of the pandemic is coming into sight. Now, in the United Kingdom, that's where we reside at the moment. We, as a country, our government are optimists, and as of next week, we will be going back to work. We will stop wearing masks, and there will be no restrictions in terms of having to prove a negative test, etc., to go into restaurants. Now, the pessimist has a different point of view. That is that although Omicron is mild, it's still dangerous enough to cause severe disease and death in some people. Now, if it does infect millions, then hospitals may be overwhelmed across the world, and it could contribute to people with long COVID or post-COVID conditions. We don't really know yet whether Omicron is going to cause a large number of people with long COVID because it hasn't been around for long enough yet. So the pessimist would say the pandemic is not over. the virus is actually rampaging across the world. And there are actually a great many scientists who feel this way too. And then there's the fatalist. The fatalist says that Omicron is like fire passing through dry grass and everyone will become infected in the end. And in the UK, the variant was introduced in the end of November. And by early January, 1 in 15 people were infected. It travels very fast. So we can't intentionally move towards endemicity. This is down to the virus and how it evolves. And so let's lift restrictions and get on with life. And let's just accept the consequences. Let's reestablish the basic order of social interactions and pre-COVID life. Again, this is a narrative that's quite similar to the one that I hear in the UK. I think I sit with a different category, and that's the realist, where we have a mixture of the kind of the pessimist, the optimist, and the fatalist all mixed together. And I think that what I would say is that actually many people will get an immunity boost in the coming months. I wonder what cost that will come with. As countries experience their Omicron wave, I think health systems will be challenged and more severe cases will occur in the unvaccinated. And so there's an inevitability that people will die from this. I think we're moving towards endemicity, but I don't think it's game over yet. And I do think we have to prepare for a new variant. So this is really my last main slide, and to think about what comes after Omicron, if anything. So the question is, are there other variants around the corner in 2022? Well, I think we should expect the unexpected and be prepared to detect what's coming next. And so sequencing remains as important as ever. We need to expect the unexpected because that's what the virus does. And I would bet, if I was a betting person, that there will be another variant to come. Now, any new variant will need to be either more transmissible or better at escaping immunity, or both. We don't know what that will be. But actually, future severity is really unknown. It's very hard to predict the evolution of virulence. It could go up, it could go down, or it could stay the same. Now, a critical point that we really need is vaccines that prevent severe disease, but also that actually prevent potentially infections. So we need lasting vaccines that prevent reinfection, because it's actually very difficult to imagine that we can keep being vaccinated every 3 months for the rest of our lives. I'd like to finish by saying that we are witnesses to our own history, which is why it makes it so difficult to predict what's going to happen next. And how we have to manage this uncertainty is something that actually has been very difficult over the last 2 years, I think, for all of us. We'll only be certain when we look back as to what exactly is going to come next. But I hope I've given you some options as to what you think might happen next and how you will actually approach that sociologically. So thank you for listening, and thank you again for the invitation. I wish you all well going into the future, and I hope that you remain well. Thank you.
Thank you very much, Dr. Peacock, for truly an enlightening lecture that's given us all a lot to think about. There are a number of questions. I'm going to take the liberty of asking you the first question myself, if I might. One of the things you brought up was transmissibility between humans and other species, and going from human to animal, within animals, and then potentially, hopefully not, but back to humans. So it seems to me that there's a great need for surveillance of that. And that entails lots of things, including investing on doing testing amongst animal populations and working with groups all over the world to keep a check on that. Do you think that strategy is a very important part of the overall mitigation plan into the future? And how do we get the countries of the world to support and invest that?
Well, I do think it is. I think that it's, I think I would call this a wild card. You know, we haven't seen a major issue in relation to the virus passing, this particular virus passing from humans to animals and then back to humans again, or even into animals. But actually, I think that we haven't really scratched the surface of it. So I think that we should be looking at the wildlife. I think that much of the animal reports of SARS-CoV-2 has been in pets, cats, dogs, and others that actually have that the infection has passed from the human to the pet. But I think that potentially the wildlife could form an important reservoir, and so I think it is important that that surveillance goes on in that regard. And I think that obviously the veterinary community are key to that.
Okay, great. Thank you. Thank you very much. All right, we have a number of questions. I'll kind of read them in order. One question said, as you mentioned, with the huge numbers of infections continuing to occur across the globe, the chances of new variants increase dramatically. Are there a few variants out there right now that are just emerging that we should be particularly monitoring?
Well, there's nothing that concerns me more than Omicron at the moment. But I think that new variants could be out there at the moment that we don't know about. And I don't want to sound alarmist, but actually many countries don't have access to sequencing. And if they don't have access to sequencing, you will only know about a variant if that infected person travels to a country that does have access to sequencing. So I think it's a really good argument for getting sequencing across the world. But at the moment, I would say that the Omicron is the one to watch. There are occasional news reports about particular variants emerging, but I think that they're not ones that I'm overly concerned about at the moment.
Okay, thank you. Carla Finkelstein, who you've met, who runs our COVID testing lab, asked, in the graphs you showed for the United Kingdom with the changes in cases and deaths, she was curious as to where vaccination started in that graph. If you can, relate that to us, you don't have to pull the graph up again. But
yeah, well, vaccination occurred. I'm just trying to think of when it started actually. So it will have been around our second wave, and so, or was it our third wave? That was a tricky question which I didn't prepare for, but we started to vaccinate about a year ago.
Okay.
And so that would have been after our second wave and before our third.
Okay, great, thank you. Zisheng asked, why does Omicron preferentially infect the upper respiratory tract with limited effect on the lungs? Is it due to the mutations or the responses of the body to this variant? What he's really asking is, do we know what the molecular basis of that is?
Yeah, okay. Well, it's likely that the virus does interact with the cells differently compared with Delta. There's early evidence on that. I think that's still the evidence that is emerging, but it may be that it's the way that it actually interacts with the cell surface and the way that it actually gets into the cell and how it's released into the cell that's important. So there's a very nice preprint out by Wendy Barclay's group in Imperial, which looks at some of those factors. But I think it has to be down to mutations. It has to be down to the genetic component of the virus. So that's what starts it. I think the key question is which mutations exactly in Omicron are leading to that change in the way that the virus is behaving and the way that the virus is interacting with the cell. But it would be down to the mutations for sure. But as I say, do go and have a look at that preprint. It's not yet in the peer-reviewed literature, but I would recommend reading that.
Great, thank you. Sahija Biswas asks, is it possible to be co-infected with two different variants? If the answer is yes, does it increase the chance of creating a new variant with both increased transmissibility and virulence?
The answer is that you can be infected with two variants, although it appears to be quite uncommon. And it's probably quite uncommon at the moment because, for example, in the UK, all you'll ever meet is, if you get infected, is Omicron, and before that was Delta, and before that was Alpha. But when there was a lot more diversity in the virus, we did detect mixed infection, and that is when two viruses can recombine, and that's another way that the virus can actually make a big leap in its evolutionary trajectory. So bits of virus can actually stick together and create a new virus. But actually, we haven't seen that to a great extent. We haven't seen that as a driving force in new variants of concern. It's something that I think is important to look out for. Is a possible mechanism by which a new virus could emerge. But co-infection is relatively unusual.
Ignacio Moore asked, is the rate of evolution of the virus changing?
Not that I'm aware of. I mean, as far as I know, then the mutation rate would be it would have 2 mutations a month in the general virus. But is it mutating? Not that I'm aware of.
Okay. Frankie Edwards asked, are there ways to model possible variants? If so, can this help countries prepare for other waves?
Mm. You're modeling unknown variants, is that correct? Is that what the question is asking?
Yeah, I'm not sure if that, yeah, it almost seems to me what you would be asking is, can you model and predict from that modeling the emergence of new variants, I would think.
Oh, I see. Okay, yeah, okay. Well, experimental biologists have attempted to model what variants might emerge that are important. And in particular, scientists have actually mutated each point in the genome of the spike protein to see what effect that would have on neutralization. And through doing that, they can predict what points in the genome could lead to problems. And so you could then model the combination of mutations and the likely outcomes from those. But actually, I don't think any models that I've seen have actually been able to be anywhere close to predicting alpha, delta, or Omicron. Actually, I think all of those three have come out of left of field. Mm-hmm. So it's interesting. It's an interesting idea that you might model what's going to come next, which is really important. But that hasn't been effective so far. I would say that each time we get a variant, we are as surprised as the last time.
Okay. We have an anonymous question. What are your thoughts on the fairly recent discussion of shifting to age-stratified COVID guidelines to balance economic stability and disease mitigation for our most vulnerable populations?
I didn't get the first bit of the question.
What do you think about discussions of shifting to an age-stratified COVID guidelines to balance economic stability and disease mitigation for the more vulnerable people?
I think that what that's referring to is where you protect the vulnerable, but you don't protect the younger people who could actually get infection and not have such severe disease. I think actually, if you're talking about going into an endemic state, that is actually what you would probably move towards, because in a situation where you're referring to something as endemic, and for example, next week when the UK moves to a period where we take off our masks and we go back to work, etc. I mean, of course, everyone can wear masks if they want to, but we're being told we can go back to work. Actually, that is effectively portions of the workforce going out into normal society again. But I don't think anybody would want to see people who are kind of being they're vulnerable from infection wanting to do that. And so I think that there would need to be a stratified view for shielding people who actually are likely to have a worse outcome from Omicron than somebody who is otherwise fit and well. So I think if we're going towards endemicity, we would be thinking about this like we do influenza. And we know that influenza is more likely to kill people of particular age groups with particular background comorbidities. And so I think it's inevitable that we would do that. And in many ways, we do that now, don't we, with shielding, with shielded people. We have different rules.
Yeah, Paul Skolnick, who's an infectious disease physician, asked sort of a question with a follow-up here. He's wondering why we haven't seen recombinants yet. And he said, with all the replication going on, it would seem there's something inherent in the virus that is limiting or preventing the creation of recombinants. Is this possible?
Yeah, we have seen, but it is possible to get recombination. We have seen it. And there are several papers, including one by Andrew Rambo, that actually does report recombination. It can occur, but it's actually not common. The biological basis for that, I don't know, actually. In fact, can I just say that there's quite a lot of things I don't know about the pandemic, because most days that, you'll get up in the morning, you think you know something, and then the virus teaches you a lesson and you feel humble again. But I don't know why we're not getting a large amount of recombination, but we certainly don't see it, which is a good thing, I think.
Okay. Yeah. X.J. Meng, a molecular virologist at Virginia Tech, asked, he said, future variants could emerge from infected wildlife species like deer through reverse zoonosis. Should we begin to vaccinate domestic and wildlife animals against SARS-CoV-2?
I think you need the evidence before you vaccinated. You need to understand the prevalence of transmission in wildlife. You'd need to sort of understand the scale of the problem. I think that to develop a vaccination program, you really would need to substantiate the need and what the action is you're trying to prevent. So I wouldn't underestimate the importance of wildlife infection for sure. But to say that the detection of a small number of cases would demand a vaccination program doesn't quite seem proportionate. So I think the message will be to understand the prevalence of SARS-CoV-2 in wildlife populations, and actually what that means for the evolution of the virus.
Yeah, okay. Ansar Ahmed, professor in the College of Veterinary Medicine here at Virginia Tech, said it's in the interest of viruses to escape immunity. So how do we address this going forward? Do you think we need a variant-specific vaccine or a vaccine that can be built from multiple predictive variants? He says there are challenges to both approaches, he imagines.
Yeah. Well, I think the vaccine companies are taking a kind of lots of different approaches at the moment. So certainly there are vaccinations being developed to Omicron, for example, specific variants. And in fact, there was vaccines developed for beta, for example. There are also vaccines being developed for sort of multiple variants. And so I think the vaccine companies are thinking about all of those. What it would be fantastic to see is a vaccine, if it were possible, to make a vaccine that actually was effective, equally effective across variants and actually targeted the whole virus rather than the spike protein. And so that's what I'd really like to see, a vaccine. Perhaps it's expecting too much, but a vaccine that actually prevents infection and targets the entire virus would be rather special.
Okay, great. All right, we just have a couple of minutes here. We'll just maybe take one or two more questions. I see Zhao Min asked, is there a theoretical upper limit for transmissibility? If so, how close is Omicron?
Well, if you look at some viruses, they are extremely transmissible. And I don't think that Omicron has reached the ceiling of its capability yet. And so when it was actually spreading very rapidly in the UK, the R value was around 5 or 6. Actually, when we look at other viruses, some viruses have an R value of 12. So I think that I would say I have actually read that this is the most transmissible virus we've ever seen on the planet. I don't think that's the case, and I think it's still got some headroom for transmissibility.
Okay. And
let's hope we don't see it, but there is headroom.
Okay, thank you. All right, I think we'll just do one more question, and then we'll let you take a rest. Kenneth Peacock said, building upon Dr. Friedlander's comments earlier, if bats are a reservoir, what are the most likely amplifier species that contributed to the current initial spillover, if it was a spillover?
Yes, that's a good one to finish on, isn't it? Because I don't think that, again, this is something that I don't think anybody can be absolutely definitive about. I mean, there's been talk about pangolins, but I think that this is going to be an area that is going to be deeply ungratifying in terms of us understanding the origins of SARS-CoV-2. And I believe myself that this is likely to have risen from an animal. But how exactly, as humans, we want to know the precise details, but the exact pathway from which it went from a bat to humans, I think we're never going to know with the current situation.
Great.
More uncertainty, unfortunately.
Oh, there certainly is a lot of that. Well, Dr. Peacock, I can't thank you enough. It's been extremely enlightening. We greatly appreciate your time and all the exciting information. And all I can say is keep up the good work, the important work that you're doing. Thank you very much.
Thank you very much. Goodbye.
Thanks to everybody for joining us tonight. We greatly appreciate it. I'm sorry we couldn't get to all the questions, but if you want to send your questions to our site, we can forward some of the ones that weren't answered to Dr. Peacock, and perhaps she would have time to answer them later. So thanks again, everyone. See you soon. Sharon, thank you so
Bye-bye.
much. It was
Bye.
really fantastic. Greatly.
Bye-bye.